It's a pleasure to have my colleague Sandeep Chaube give the next talk. Sandeep has been in IMSc, a faculty in the computational biology group for close to 2 years now. He did his PhD in Brandeis, subsequently postdoctoral stints at Harvard and MPIPKS and Dresden. He's a biophysicist and he has varied interests. So welcome Sandeep, and thanks for accepting our invitation and looking forward to your talk.
Thank you. Again, I envy those who gave their talks before lunch. It's just much harder to talk, I mean, when you are full, basically. I would like to thank Arijit and Ajay also for organizing this wonderful, this network biology day, as they call it. It's been a fascinating day, so I learned a lot, and most of the things are quite new to me since I don't work on some of these topics. The work that I'll talk about is a theory experiment collaboration between my group and a group of, the group of a friend who does experiments. So I don't know where network comes into this. So it may be at the last, you'll see the last slide where there is some hint towards it. Okay, so it's a very physical biology kind of story. It's a very simple story, as you'll see. So we'll talk about something, the title of the talk, as you can see, it's called a multi-component rendezvous of proteins at actin barbed end. This rendezvous, this word, of course, I like a lot because there is a restaurant in Pondicherry which has the same name, and they make some amazing food. Okay, so first of all, let's take a look at some of these different organisms that evolution has essentially thrown at us. On the left-hand side, you have white blood cells. In the middle, you have amoeba, and on the right-hand side, there is a single cell. It's a fish cell that we have. And as you can see, they come at different times during evolution, but all of them have this common thing, which is cell motility. So cell motility is a conserved phenomena in evolution that shows up all the time, and something that is fundamental to biology in many ways. And here is a very simple, one of the videos that I showed, this example, and this is taken from this particular paper. And if one zooms in and sees the protein that is responsible for cell motility, these are actins. And as you can see, it's an electron micrograph image, and it really looks like a mesh. Right, so these actin can be in two different forms. This particular protein, it can be in a filamentous form or a monomeric form, and they can form this really branched mesh-like structure. So, and at the core of it is a single actin filament. And the focus of this talk will be mostly this at the filament level. One of the key features of this process is that you have these really tiny monomeric, like nanometer scale proteins that end up achieving something which happens at the micron level, right? So there is some sort of collective behavior that is at play. So all these tiny molecules come together in a magical fashion to give rise to very well coordinated movements of cells. Now, here is a very simple it's a very beautiful movie that I like. So it's a particular cell. As you will see that the cell is moving, and they have tagged the whole actin pool. So they tag the monomeric as well as filamentous actins. And as you can see, the cell is moving in a certain directions, and at the tip of the cell, when the tip of basically the movement, the direction of the movement, you will see that these actin filaments are pushing against the wall, thereby this whole cell is moving. But while you are pushing against the wall, you have to also replenish the monomeric pool, right? So you are creating these filaments that are pushing. So the filaments, the size of the filament also has to be taken into account by the cells, in the sense that they have to constantly kind of give rise to monomeric actins, which they can use again to polymerize at the tip, so they can move against the wall. Right? And so this is kind of a typical actin structure, and the way it really happens is that you have these monomers that come and bind to the barbed end, the plus end, as I call it, and thereby the actin can polymerize in that direction, and then it can also depolymerize from the pointed end, so that it can lose these monomers. Because you have to do all these things, the cells have to achieve all these things with some amount of monomer pool, and they have to manage the pool in a manner that they can achieve all these complicated kind of motility. So one of the key questions is that how do cells integrate these various kind of proteins, the activity of various proteins which are responsible for this. So far I just told you that I've been telling you about actins, that there are monomeric actins and there are filamentous actins. But there are other subsidiary proteins that are also extremely important. They come together and allow the tuning of polymerization and depolymerization. So if I think of this really fundamental process of filaments which are growing or shrinking, the growing and shrinking processes are really tightly regulated in cells through the action of many proteins, which goes to about hundreds. And they are called with different names like branchers, blockers, sequesters, and so on. And for our purposes, that's not too important. What is really important is that cells are integrating the action of many proteins to achieve what happens at the micron scale that you saw. Okay, so first we'll focus on 3 different proteins, and we'll try to understand how these three proteins, the activity of these 3 proteins are integrated. Now these are twinfilin, profilin, and cofilin. Now they are known to be depolymerizers. So all they do is that they go and bind to the barbed end, the tip of the filament, and then changes the rate at which the cell is losing monomers. Sorry, the filament is losing monomers. That's it. So this is all these 3 proteins do, right? At an individual level, the action of these proteins are very well characterized. So decades of biochemical studies, they have figured out what is the rate at which they bind, what is the dissociation constant, and at what rate they depolymerize actin filaments. And we ask a very simple question, which is that how do they together work, right? So together, is it some sort of an additive effect that if I know the individual effects, can I predict what's going to happen together, right? That's really the question. It's a very simple question, but something that accounts for the actual situation of a cell, where you really don't have single proteins, which is what people typically focus on. So if they are acting at the same barbed end, then do they cooperate or they compete? Right? That's kind of the first question one can ask. If 2 proteins are coming, I mean, if we are really interested in figuring out how 2 proteins act on the actin filament barbed end. Now, the typical approach to answer this question is to visualize the proteins live. Like, so you really try to see how the proteins go, and you label them with different colors, and you see how they go and bind to the barbed end, whether they can simultaneously occupy the barbed end or not, and so on. So that's how you really figure things out. But an alternative approach, which is what I'm going to talk about, is to come up with a theory experiment dialogue, where essentially you make hypotheses about the systems, you turn them into simple mathematical models, you compare them to the data, and then you see if the model really works. So and then you and the models, of course, are falsifiable, so that you can actually compare them rigorously to experimental data. And then if it doesn't match, then you throw it away, and then you try to see how to modify the model to actually account for the data and so on. And you'll see what I'm talking about. Now, the reason I had it as a microscope, the way I see it is that it's an alternative microscope for an experimentalist who can really kind of, rather than visualizing them, use this other method to kind of decipher the actual mechanisms of such multi-component regulation. Now, the simple, I'll just briefly tell you about the simple experimental setup they have, which is basically they have a microfluidic chamber, and they can now grow actin filaments there. So the actin filament is attached to the cover slip, and then you can flow in any proteins from the left-hand side, and then it can go away, and so you can basically do a lot of manipulation with them. And the end result is a movie like this, where you can see each actin filament is attached to the cover slip, and you can see that it's growing. In this case, it can also shrink. You can see their whole dynamics. So it contains a wealth of information about the underlying processes. And the question is, how do you actually use it to learn something about these molecules. So first of all, what we do is that we characterize the single proteins, which is something that people have done earlier also, but this is kind of for the sake of completeness. So what we have is, on the x-axis you have twinfilins. You add twinfilin molecules to the system and systematically change its concentration, and you see how the depolymerization rate changes. Now, as you can imagine that, the curve basically simply goes up and then kind of saturates, which is what you would expect at high concentration. The filament is always occupied by a twinfilin molecule, so that's why it saturates, and then that gives you the corresponding depolymerization rate at the protein twinfilin bound state, and we'll talk about the quantification. Similarly, you can do the same exercise for other proteins such as profilin and cofilin, right? Now, you can use a very simple model to actually understand the data, which is that you say that, okay, your filament can occupy two different states, and the two states have certain weights, right? For what fraction of time they'll be occupied is given by simple stat mech that you can write down, and here the Cp is the concentration of the protein and Kdp is the dissociation constant of the protein. That's it. And then from there you have some rate of depolymerization from these two states, and you can actually compute the average or experimentally observed depolymerization rate, which is just nothing but the product of the depolymerization rate and the corresponding occupancies and the sum of it. That's it, right? Turns out, and this is the final expression, turns out you can actually learn about the underlying rates. All you do is that you fit the data to this model, and you can extract Kd and the other rates, right? Rate of depolymerization of these proteins. So we can extract these parameters that we will be working with. Now, what we do in order to understand how three proteins would work, first we take two proteins at a time. So you have three such pairs, and we consider two broad classes of models. The first class of model is a competitive model. So in other words, as I said, so if they are binding to the same site, the null model would be that they cannot occupy the same site at the same time, right? That's the simplest kind of model. And then it can also happen that they can cooperate, so they can together occupy the barbed end and then thereby facilitate depolymerization. Now, the competitive model essentially looks like this. You have three different states. Either the filament is bare, or it binds protein A or protein B with these different weights, right? And the corresponding depolymerization rates. Now you see for every protein pair, we already know these rates and the KDs because we have already characterized them through the single protein assays. Since we know that, we can actually make predictions about what's going to happen if I add both the proteins and tune the concentration of one of those proteins. This is the other model that we'll talk about. The other model allows for the occupancy of both the proteins simultaneously at the barbed end, right? So that's the difference between these two, and it has a corresponding depolymerization rate. Now, if I so these are the two classes that we'll talk about. Now, if I just say that, look, you have protein A and protein B. This is how they look like individually. If it's a competitive model, you would see something like this, which is the black line. Now you see there is an interesting feature here, which is that at low concentrations, the proteins actually work in an additive manner. The reason is that although they are occupying the same site, they're competing, at low concentrations, the competition is not really felt by the individual protein. So essentially, they end up acting as adders. But at a higher concentration, they really start feeling competition, and then that's why it becomes subadditive. On the other hand, if I look at the cooperative picture, it can be additive in the beginning, or even later, or it can be super additive. So these are the two broad regimes that you are going to see. Now the question is, what happens if we add twinfilin and profilin? Now this is the depolymerization rate that you observe just with twinfilin. Now if one adds twinfilin plus 100 micromolar of, one changes the concentration of twinfilin, but also adds 100 micromolar of profilin. In the presence of the profilin, one sees what happens. And as you can see, the competitive model really explain. I mean, this is a prediction from the model, and it matches really well with the data. So what it tells you is that they are really competing. Similarly, if one looks at, if one tunes profilin concentration but keeps the twinfilin concentration fixed, it actually matches well as well, right? So what it tells you is that a competitive model can explain whatever is happening with twinfilin and profilin. Similarly, you can look at twinfilin and cofilin and see something very similar, that a competitive model can explain the data really well, right? Again, this is not a fit, it's just a prediction coming from the individual parameterization of the proteins. So then we ask what happens with cofilin and profilin. So it turns out that if you just change cofilin, this is the black line. Now you change, you add again. I mean, you change cofilin, but in the presence of 100 micromolar of profilin, and the overall depolymerization rate increases. But then this is the prediction from the competitive model. What you see is that initially the red data point is below the competitive model prediction, but then it consistently is higher than that. So what it tells you is that it hints towards some sort of cooperation between the proteins. So in other words, you cannot have such high rates of depolymerization without their simultaneous presence at the barbed end and overall increase in the depolymerization rate. So then what you see is that from the depolymerization, the competitive model, all we did was to say that, look, we assume that the KDs do not get affected by the presence of the proteins. In other words, they bind with the same affinity. It's just that when they are together, we allow that state. So when they are together, there is a rate at which they depolymerize. It turns out that rate is much higher than the individual rates. It's about 12 subunits per second, right? It's much higher than the other ones observed. So in other words, they can really occupy the barbed end together and increase the overall rate of depolymerization. So, and you see that the same thing if you are tuning the other protein and keeping cofilin constant. So you see that it's basically consistent with the model. Now then, what you can actually do is that now that you know the pairwise interactions, we can ask the question, if we add all the three proteins together, what's going to happen, right? Because we know individually how the pairwise how they interact. So all that you have to do is that you have to combine all the different possible states that are allowed by these pairwise interactions. And you already know the different rates. You can make a precise prediction of what's going to happen, right? Turns out that if you look at the black data points, which is that you are just changing cofilin concentration and you are observing depolymerization rate in the presence of twinfilin. Now you add also 100 micromolar of profilin. So you have both twinfilin in the solution and profilin in the solution, and you are tuning cofilin concentration. You see, that's the data. That's how the data point looks like. Now you compare it to the predictions from the model, which was built on pairwise interactions, and this is how it looks like. The model actually predicts that the overall depolymerization rate should increase in the presence of the other two proteins, but it actually goes down. What it really tells you, what it implies is that there are higher order interactions in the system. So what do I mean by that? Essentially, if I'm trying to predict what's going to happen with three proteins, I cannot really reduce them to single or pairwise interactions at all. Something which I like to call more is different, taken from Anderson's famous physicist Anderson's paper. So and this is consistent with the other data set also. You can tune other proteins keeping the two other fixed, and this is a behavior that persists. So now some connection to networks. So what it really basically tells you is that this kind of picture, if I look at gene regulatory network or protein-protein interaction network or microbial ecology networks, what they are built upon, that typically they are built upon this premise that if I know gene A and gene B, if I figure out the interaction between gene A and gene B, and if I figure out the interaction between gene A and gene C, I should be able to build a network based on these pairwise interactions. They always assume that you have some pairwise interactions that is sufficient for you to build the whole, look at the whole network, or kind of predict the behavior of the whole network. But that is not quite true. right, in this case. What it tells you is that you really have to care about how to build predictive models with them, because there are, like in other words, if you add 3 proteins together or 4 proteins together, you cannot really explain them from pairwise interactions. You need to account for higher order interactions. This is something that pops up in many different systems and complex systems. In particular, there are recent data even in microbial ecology where people have seen that you cannot explain the behavior of some number of microbes just by looking at pairwise interactions. So higher order interactions are super important. So I'll just end with this. And the basic conclusions is that you can actually use a simple theory experiment dialogue to unravel mechanisms of multi-component regulation. The other thing is that twinfilin competes for binding with profilin and it should be profilin. And then effect of 3 proteins cannot be explained by simple pairwise interaction. So these are really the key take-home messages. This is Shashank from Emory University. He's the PI of the lab, and Ankita is a PhD student who did most of the experiments. And I would also like to thank DBT as well as DAE for all the grants and everything. Thanks a lot.
Nice talk. I was wondering, how are you exactly modeling the competitive and cooperative interaction? Is it through Hill coefficients or some other means?
No, nothing like that. You just, so essentially you have this, so it's a very simple grand canonical ensemble that we use. So essentially you say that chemical potential is fixed, and you are all, I mean, of course, it's in contact with a thermal bath. Then you can just write down the partition function. Now here we are writing in terms of KDs because that's something people report. So it's easier to, chemical potential is harder to measure and so on. So that's it. And so just simple StatMech at the mean level, that's all.
So how would competitive differ from cooperative in terms of that?
Yeah, so cooperative will have a few more states. The cooperative will allow for a state where simultaneously both proteins can bind the barbed, other than the competitive where you can just have 3 states where individually they're allowing. So, and then that state will have some corresponding rate of depolymerization, the additional state in the cooperative model.
So you're also specifying the energy drop because of the bound state or something?
So yes, so when they're together, we assume that they bind with the same energy. So we do not assume anything different about them. That's because if you add that makes the model more complex. We wanted to keep it, just wanted to see how much more complexity we want to add to account for the data. So the only additional parameter is this rate of depolymerization from this.
So when you model pairwise interactions, you assumed either competitive or cooperative. So when we try to model the 3 protein interaction, can the same philosophy be used, like the pairwise interactions being competitive or cooperative?
So if I follow you correctly, so exactly that's what we do. I mean, basically what we say is that, look, we know how the 3 different pairs behave. So we know twinfilin competes with cofilin and profilin, and then cofilin and profilin can bind the barbed end together. So we are allowing all the different states that account for these facts. So essentially we are saying, look, since we know that these proteins compete, so they cannot be together, and we know that cofilin and profilin can be together, so we allow that state. That's it. And we know all the other rates and all the parameters that describe this because from the pairwise interactions. So then you try to see if you make a prediction, does it match with the data? That's what you ask.
Maybe let me put it this way. So we know 2 proteins, they're competing.
Yes.
Now when the third protein comes,
yes.
So it may not be guaranteed that it is still in the competitive.
Exactly. No, exactly. That's really what's happening, right? I mean, you are so the whole point is that you want to slowly build. It's a bottom-up approach that you have. So we just assume that, look, let's say that we can explain it from, let's assume that we can explain it from pairwise interaction. Turns out that you cannot.
You cannot. Yeah,
so when the third one comes, something weird happens.
Right.
And so far, I mean, they are doing some experiments, but it seems like when the third one comes, they actually can form some sort of a complex. And then essentially that reduces the effective concentration of some other protein. But whatever happens, it's not like they're independent. So there is some sort of emerging property that comes up. That is really the main point.
Okay, thank you.
So if you have 3 of them together and add that to the network in addition to 1 and the 2, that should be able to explain this data.
Exactly,
right?
exactly. But at this point, see, that's like if you add another parameter, of course you will explain the data, right?
Okay.
The question is, how should we, like now, if I mean, as I said in the beginning that there are hundreds of proteins. Now, how should we even approach this problem is not very clear to me. Like let's say you want to predict with 5 proteins or 6 proteins, and we can do those experiments very easily in the lab. Now, but how are you going to build such a thing? Are you going to just follow the same procedure, but then you have to keep adding parameters? Is there a better way to do it? It's not clear.
But with the 3 you can, right?
With the 3, I can, yes. With the 3, of course we can. But we are trying to see if there is an alternative approach to it, and maybe there are some ideas from graph theory that we can employ. Which one?
The filaments growing in the microscope, right?
That one. This one.
Yeah. So they are just growing, right? Nothing else is happening there, or what?
This one, yes.
Yeah.
So this one is when you just add monomers, so they're just allowed to grow. Basically.
Right.
They don't have any other proteins. But of course, I mean, for different proteins, different things happen.
Sure. Yeah.
Any further questions? If not, let's thank Sandeep again for this wonderful talk.
Thanks a lot. Yeah. Thank you very much.
Sure.